So now we're going to talk about some systemic dimorphic fungi. And these fungi are able to cause infection in immune competent. And this is important because it shows just how good of an immune response you have against fungus. Because usually with minimal disease. So still, in the case of our systemic dimorphic fungi, it really is the immune compromised that are going to have trouble with these. But these are going to be diseases that can cause infection and immune competence, so that means our immune compromised can get very severe diseases with these. So one of the big things about these fungi is for avoidance if you are immune compromised. So they're systemic dimorphic fungi. That means they are a yeast at 37 degrees. So this is thermal dimorphism. And they're going to be a mold at room temperature, about 20 to 25 degrees, somewhere around there. Now we're going to talk about several of these that are very common human pathogens, and we're going to talk about just where they are. A lot of the endemic mycoses are going to be in certain locations. And so if your patient has never even traveled to those locations, it's not something that you could consider them having. But if they have traveled to certain locations and they have a disease that's presenting similar to a fungal, you can start thinking about one of these guys. All right, some facts. So we have our domesticated animals that can acquire blastomycosis by inhalation of spores. The transmission of these is not from pet to pet or pet to human or human to human. It's basically an inhalation of the spores from the environment. And oftentimes spores can travel a long way. The infections with Blastomyces and Histoplasma are associated always with a moist soil in river valleys. Infections with Coccidioides is going to be with dry soil in deserts. So again, about location. If you had somebody that was visiting the Ohio River Valley, you would not think that they got infected with Coccidioides. Neither Blastomyces or Histoplasma is isolated from the soil, so they're very hard to find in the environment. These three fungi are all dimorphic, and all of them are going to have an initial pulmonary disease. This is often mistaken for tuberculosis, and one of the reasons is because they are very slow and insidious in onset. And so you might have traveled to the Ohio River Basin, picked up this mold, and months later present with a disease, and you didn't even associate it with your trip to the Ohio River Basin, for example. And so they're going to be very slow. They are molds. They have very slow doubling times, and so they're going to be thought of more as potentially other chronic infections such as your tuberculosis or lung cancer. All right, so these guys are going to be found in certain areas, as I mentioned. So I'm going to sort of blow this up, and we'll talk about a little bit of these. So you can see in the United States, we have a group that is this orange. And so we can have in this orange Blastomyces dermatitis, Histoplasm capsulatum, variety capsulatum. And so that's going to be found in the sort of the Ohio River, Mississippi River area. We also have this green zone over here in our southwestern area, and that's going to be our Coccidioides. Our Coccidioides immitis, Bodaci are going to be part of that. We also do have a lot of interaction, of course, with other areas of the world. People travel a lot, and so in our Central America regions of the world, as well as our northern regions of South America, We're going to have our Coccidioides brazilis. We have our Histoplasm capsulatum is also going to be found there as well. In South America, this purple is going to be another area with Histoplasm capsulatum. So any of those areas, if you have people who are traveling to, can potentially have Histoplasm capsulatum. We have the variety Duboisi in Africa. So if you have somebody traveling to the Ivory Coast or the Western African coast, they might come back with another variety of Histoplasm capsulatum. And so again, it's all about location with these guys. So let's talk about each of these. So we have our Blastomyces dermatitis, and this is going to cause blastomycosis. And this is going to be, again, endemic in the United States. We have our southeast, eastern states, southern states, especially those that have water. So the Ohio, Mississippi River Basin, our Midwest, all the way up to Canada can get infected, you can get infected with blastomyces. The outbreaks are going to be occupational, somebody who works in those wet areas, or recreational. Typically, you're going to have more men are going to be affected with this because they do more outdoors things, hunting, fishing, camping. And again, spread to spread from human to human does not occur. So you actually have to pick up the spores in the environment. So the life cycle is going to be, again, a mold in the environment. So it grows as a septic hyphae, and it's going to be parasitic yeast inside of us. So inside of our bodies, it has these spores that will germinate and make these budding yeast, and these are diagnostic for blastomyces. You can see that blastomyces can have an influence from a dog, for example. So pets can also get infected, and then they can release the spores in the environment, and then humans can inhale those spores, and they can get infected. So you're going to have our clinical disease, which I'll put up here because I have more room. Our clinical disease is going to be mostly asymptomatic. So again, most healthy people can control this. It's not going to cause a problem. For people who are immune compromised, you're going to get a pneumonia. It can disseminate to the skin and subcutaneous tissues. Sub-Q. It can disseminate to the bone, central nervous system. And so again, it typically is not going to cause a problem in people who are immune competent, but for immune compromised, the disease can get pretty widespread. The diagnosis is the detection of yeast cells, so you're looking for these budding yeast. You can also, there is an antigen detection test for this particular fungal agent, so you can use urine to look for the proteins from your blastomyces. Now it's going to grow very slowly, so it grows as a mold. It's going to take 2 to 4 weeks, and it grows as a yeast in about 3 to 5 days inside of an incubator. So you can put it at both locations and see if you get growth of blastomyces. The next one is Coccidioides. Now Coccidioides immitis and podaceae are going to cause coccidioidomycosis. The difference between immitis and podaceae is only differentiated by molecular methods, so typically you would treat them both kind of the same. Now they are going to actually form a spherical inside of us. So we talked about those sporangiums in our introduction, in which you had sort of this nice little compartment that you filled with spores. Well, that's sort of what Coccidioides does inside of us. It makes a spherical, and it stuffs a bunch of endospores inside of this spherical. And this is going to be key in the diagnosis of this, looking for these sphericals in human tissues. It also has a unique mold form. It does not form spores at the end of its hyphae. Instead, it's going to have septate walls, and then it's going to disintegrate the cell walls, and it leaves these funny barrel-shaped cells. And these barrel-shaped cells, which I'll show you in just a moment, are arthroconidia. And arthroconidia are its infectious spores. So in essence, when we showed you the septate hyphae, you have this hyphae, this long hyphae, and it had these individual cells. Well, basically, they just break off. And you get these barrels. So these are pieces of the hyphae. And these are going to be those arthroconidia. Right, so that's what this is, arthroconidia. Arthroconidia are infectious spores, even though they're really just chunks of hyphae. They are going to help protect the DNA in the nucleus, and they can travel for hundreds of miles. So you might be walking around in one location, and hundreds of miles away there was a Coccidioides that released its arthroconidia, and now it's infecting you. So Coccidioides, again, it is going to have a mold on the outside of our bodies, and on the inside it's going to be considered a yeast, but it's a spherical. So here we inhaled our pseudo hyphae, and then we're going to, or our arthroconidia, we inhale our arthroconidia, which are chunks of its hyphae. It just breaks off these little chunks and becomes our arthroconidia. And then inside of our bodies, these arthroconidia are going to form into yeast cells, which then are going to quickly form these sphericals. And these sphericals are going to be filled with spores, and they just keep growing until the spores release. And then they continue to do this over and over again. Now, the epidemiology is that it's going to be endemic to the Southwest desert, also northern Mexico, in some areas of Central and South America as well. And again, the spores are going to be able to be disseminated from the wind, those arthroconidia. And most people are going to be asymptomatic. There is a risk for a more severe disease associated with dissemination in certain groups of individuals, including those with Filipino, African American, and Native American, Hispanic descents. It's also important to note that individuals who are pregnant, who are in their third trimester of pregnancy, are highly susceptible to this, as well as just those people who are immune deficient. So Coccidioides, valley fever is going to be more common in pregnant women, and a lot of times people who live in the Central Valley of the California, they know very well Coccidioides because of valley fever association with the third trimester of pregnancy. Again, no person-to-person spread, and lab infections can occur. So this guy grows very quickly in the lab, so you have to be very careful if you're handling a potential case. Again, the diagnosis is going to be these sporulating spheres in tissues or in the sputum, the coughing up. The mold is going to grow very rapidly, 1 to 2 days, so that's very different. It's a very highly replicating mold. Most molds take 1 to 2 weeks, so this is very fast. And so because it is the mold form, they have a lot of spores, those arthroconidia that they form, and so you can get infected clinical technician can get infected with your Coccidioides. Now, your next endemic mycoses are going to be your Histoplasm capsulatum. Histoplasmosis is going to be caused by either the capsulatum or the Duboisi variety. And Duboisi, again, is in Africa, and the capsulatum variety is in eastern United States and Latin America. So again, it has to do about where people are. You're going to have very similar forms of yeast. Capsulatum has a little bit more thinner walls, and Duboisi has thicker walls. But apart from that, they look pretty similar. Now, just again, you have your Ohio, Mississippi River Valley. Mexico and Central and South America are where most people in the United States are going to get exposed. They're going to have soil with high nitrogen content, so this means bird and bat droppings. And again, similar to all our other endemics, person to person does not occur. Now, the Histoplasm capsulatum, again, is going to have a mold form on the outside and a yeast form on the inside. And it has this multi-buddying yeast that is shown here. It looks like a little turtle. You're going to inhale the spores, and the spores are going to germinate in the lungs, and these little turtle-looking yeast are going to be diagnostic for your histoplasm. Now, the clinical disease, of course, is going to depend on the immune status again. So it goes back to if a person is immune compromised, dissemination and infection of multiple tissues is more likely. An immune competent is going to be asymptomatic or flu-like symptoms. You usually have this pulmonary infection. If you do present with it, is going to get resolved within a few weeks. But if you are immune compromised, it can disseminate. Diagnosis, again, is going to be on the microscope and culture of respiratory material. You can use serology except for people who have AIDS. So serology is not very useful in AIDS patients, so it would not be used here. You also want to look for the presence of those yeast in the urine or serum, and this is going to indicate dissemination of this particular fungi.